Hello, everyone. My name is Lauren Velie, and I'm a member of the Velie Lab at the University of Massachusetts Amherst. Today, I will be presenting on my project, Unraveling the Role of AKR4C proteins and nitric oxide homeostasis in Arabidopsis thaliana. This research is supported both by an NSF grant and a summer undergraduate research fellowship from the American Society of Plant Biologists. So the long-term goal of this project is to broadly understand the role that these AKR4C oxidoreductases play in nitric oxide homeostasis in Arabidopsis thaliana. So the general approach, we have two main approaches, first of which is to generate an AKR4C knockout line or multiple lines by a CRISPR-Cas9 mutagenesis, and secondly, to characterize these AKR4Cs and related proteins in vitro. So nitric oxide itself is a very crucial signaling molecule that's involved in many physiological processes, both developmental and in response to stressors. So nitric oxide leads to post-translational modifications such as S-nitrosylation and nitrosylation on a wide range of proteins, which then alters the protein activity. So the proteins that the signaling molecule interacts with are involved in a wide variety of processes such as shoot development, immune response, seed germination, spiral closure, fertility, and root growth. So this is a ubiquitous and extremely crucial signaling molecule that has a wide range of effects throughout the entire plant. So our main question for this project is what is regulating nitric oxide levels in the plant, and specifically what enzymes are regulating these processes. So the major enzyme that is well characterized to regulate endogenous nitric oxide levels is reductase, or GSNOR. So the stable and bioactive form of nitric oxide in the plant is known as GSNO, which is then metabolized by various enzymes, beginning with a step catalyzed by GSNOR. So by metabolizing GSNO, this protein is known to regulate endogenous levels of nitric oxide and is thus vital to the wide range of physiological processes that nitric oxide is involved in. Another important characteristic to remember is that GSNOR uses NADH as a coenzyme. So on the left of this slide, we can see some phenotypes that are characteristic of the loss of functional GSNOR in the plant, most of which are pretty extreme. So from these characteristics, we know that GSNOR acts in a variety of vital processes. But we're asking the question of what else is going on, what other proteins are influencing nitric oxide levels. So along this line of questioning, our group completed proteomics, comparing the expression of proteins in wild type and those lacking GSNOR. So like all cellular systems, the homeostatic regulation of GSNO is maintained by a variety of enzymes. So proteomics analysis is a powerful tool that we can use to identify additional enzymes that are responsible for this regulation, especially from plants lacking the prime regulator GSNOR. Here's a volcano plot showing the differential expression of AKR superfamily proteins in the GSNOR lacking plants. So as can be seen to the left, the 4C subfamily, the C9, C8, C10, and C11 we see here are especially upregulating these plants. C8 and C9 are upregulated in leaves, and all four are upregulated in pistils, the plot of which is shown here. So what are these Aldo-Keto reductases and what roles could they play in nitric oxide metabolism? So AKRs as a superfamily are known to be monomeric NAD pH dependent oxidoreductases. They are found across a wide range of organisms similar to GSNOR, including mammals and plants, and react with reactive carbonyls to form alcohols during periods of stress. So AKRs have been shown to metabolize GSNO, and our group's collaborators at the University of Bologna in Mirko Zaffagnini's group has also shown that NAD pH-dependent GSNOR reduction increases in Hop5 plants. Those are the plants lacking GSNOR. So these two factors establish an extremely strong link between dysregulated GSNO levels and AKR activity. So an important characteristic of these proteins is the high degree of similarity shared within the subfamily. Here's an alignment of all four members where I've highlighted the structures important for substrate binding and catalytic activity. And we can see that there is at least some degree of conservation and even the divergent loop structures that are important for substrate binding. So the similarity ranges from 60 to 80%, depending on what you're comparing. So these members of the AKR4C subfamily have very similar sequences and are known to react with a broad range of substrates. So it's likely that they exhibit redundancy in some functions. In order to observe any potential phenotypic differences, it's necessary to knock out all four of these proteins. So our approach utilizes CRISPR to target and introduce 2 double-strand breaks in each gene to generate a large deletion. Here's an overview of the 4 targeted genes that the large deletions have been generated in. As you can see, they range from 63 to 360 base pairs, all spanning exon regions of the genes. So why do we use CRISPR and not single mutations? Well, 3 out of the 4 genes of interest are located extremely close to one another on chromosome 2, which makes the generation of multiple knockout mutants extremely difficult when using a genetic cross approach. So we introduced this CRISPR-Cas9 machinery into the plant via floral dipping into an agrobacterium culture that has been transformed with the transformation vector that contains all of the CRISPR and guide RNA machinery necessary. So we can then screen for positive transformants through growth on selective media or through individual seed selection via an RFP that is encoded in the plasmid as well. Next, these plants go through a series of crosses and back crosses to ensure a homozygous deletion genotype, as well as the loss of the CRISPR machinery and thus any background mutations that have occurred when CRISPR was still present and active in the plant. As you may notice, it takes 6 to 8 weeks between each of these generations. So this is a time-intensive process, but generates very stable plant lines that we can work with in the future. So next, the way that we identify these large deletion mutants is through PCR genotyping. So at each step in the workflow, when there is segregation between wild type and heterozygous and homozygous plants, we aim to identify and move forward with the recovered homozygous plants to ensure no functional copy of the gene is present in the plant as we are testing them. So a variety of these plants have been recovered from this process, with the C11 deletion mutants currently on their 2nd round of self-pollination to recover homozygotes from the first back cross. All other lines, including the remaining C8, C9, and C10 single deletions, as well as double mutants, C8, C9, and C9, C10, have been recovered from their final round of backcrossing and will be available for further analysis and use in the introduction of additional mutations, such as a C8 mutation into the C9, C10 double mutant, which we can then cross with the C11 to obtain the final quadruple knockout. So our second main goal of this project is to analyze these proteins in vitro. So we have selected 3 related AKRs, so phylogenetically related to the AKR4C proteins highlighted in red, for further study. So with these selected proteins, we can examine how unique the GSNR-like activity is to the AKR4C subfamily. So in order to answer the question, is the GSNO reduction activity observed in the AKR4C members coincidental to their upregulation in Hot5 plants? So we also do this in order to ensure that the increased NADPH GSNO metabolism is due to the AKR4C subfamily specifically, rather than broadly related to the existence of an activity of AKRs within the plant. So I have expressed, purified, and assayed 2 out of the 3 related AKRs and have found that AKR2A4, the least closely related, so the purple one, actually exhibits no GSNO metabolism in vitro. AKR4C18, the most closely related AKR, has not yet been purified, and there's currently no published evidence of reactivity with GSNO. However, AKR4B9, the green highlighted related AKR, exhibits GSNO reduction activity actually similar to that of AKR4C8 in vitro. So here are the kinetics data I generated for AKR4B9. I measured the reaction rate through the disappearance of GSNO with different concentrations of both protein and GSNO and found that the kinetic parameters were actually similar to that of AKR4C8, detailed in the table below these figures. So this is an interesting result, especially considering the fact that this protein is downregulated in GSNO are null mutants. However, it is very exciting to collect brand new data on this enzyme. It will be interesting to see how the most closely related AKR, AKR4C18, behaves when successfully purified. So, in summary, first off, a great deal of work and progress have been made in each aspect of this project. But these AKR4Cs represent previously unrecognized enzymes with the ability to metabolize GSNO, the major nitric oxide reservoir in plant cells. And so plants carrying mutations in all 4 AKR4Cs are currently in development and will be available to test their specific roles in nitric oxide homeostasis in vivo. And then finally, the possible role of these other AKRs, these related AKRs in Jason reduction, is actively being tested. Thank you for watching.